You know, we always hear about our DNA as this fixed blueprint for life, right? A static code that makes you, you. But what if I told you that's only half the story? The real action isn't just in the code itself. It's about which parts of that code are being used at any given moment. Today, we're diving into that. We're going to see our genes in action. Okay, so let's jump right in with a classic question that kind of blows your mind. Humans and chimps, we share something like 98% of our DNA. So, if our genetic blueprints are that close, why are we so different? The answer, it turns out, isn't just about what genes we have, but which ones are actually switched on.
And that whole process of switching genes on and off, that's called gene expression. The best way to think about it is like this. Your entire genome is this massive, massive cookbook with thousands of recipes. Gene expression is the act of deciding which recipes to actually cook today. You see, it's not just about having the recipe for, say, an eyeball protein. It's about turning that recipe on at the right time and in the right place. That's where all the magic happens.
All right, so here's our game plan for this explainer. We'll start with that genetic puzzle we just talked about. Then we'll look at the tech that lets us take a snapshot of our genes. After that, we'll see how we turn that picture into actual data, clean it up to find the real signal, and then pinpoint the most important genes. And finally, we'll see how it all comes together to reveal these incredible biological patterns.
So how in the world do you take a picture of thousands of genes all doing their thing at once? For a long time, you just couldn't. But then, this revolutionary piece of tech came along called the microarray.
The amazing thing about a microarray is its sheer scale. I mean, instead of looking at genes one by one, it lets us measure the activity level, the expression, of literally thousands of them all at the same time. It's like going from listening to a single violin to hearing an entire orchestra play a symphony. It was a total game changer. Okay, so how does this thing actually work? Let's break it down. First, you grab your two samples. Let's say some healthy cells and some tumor cells. Next, you pull out the active gene messengers, this stuff called mRNA, and you tag them with fluorescent dyes. We'll make the healthy ones green and the tumor ones red. Then, you mix them all together and wash them over the microchip. Now, this chip is covered in thousands of tiny spots, and each spot is a probe for one specific gene. The tagged mRNA finds its matching spot and sticks to it. And the final step? You hit it with a laser. What you get is this beautiful glowing pattern of red, green, and yellow dots.
Okay, that glowing image is just wow. It's incredible to look at, but scientifically, it's just the beginning. The real journey starts now, turning this beautiful picture into cold, hard numbers we can actually work with.
This is where we hand things off from the biology lab to the world of data science. You see, every single one of those glowing spots represents a gene, and how bright it is tells us how active that gene is. Our job now is to translate all of that visual info into precise numbers. We have to measure the brightness of every single spot.
This process has a name: feature extraction. A computer program first figures out the exact location of every spot on that grid. Then, it identifies all the pixels that make up each spot and measures how bright they are. And, this is key, it also measures the area around the spot to figure out any background noise, which we'll need to deal with in a sec. Finally, all that pixel data gets converted into raw numbers, a specific intensity value for every single gene.
So now we've got our numbers. Awesome! But they're messy. The raw data is full of all sorts of experimental gunk and biases that can totally hide the real biological story. So our next job is absolutely crucial. We have to clean the data. It's all about finding that true biological signal hidden inside all the noise.
First up is something called background subtraction. You see, the glass slide itself has this faint, non-specific glow. If we don't account for that, it could make a gene look way more active than it really is. So we simply take that measurement from around the spot, the background noise, and subtract it from the spot's total brightness. What's left is a much, much cleaner signal.
Next, we do something called a log transformation. Now, I know that sounds super technical, but the reason for it is actually really simple. Before you do this, the data is kind of weirdly skewed. A gene doubling its activity looks like a huge change, while a gene cutting its activity in half looks like a tiny change. But biologically, those two events can be equally important. So the log transformation rebalances everything. Now a twofold increase and a twofold decrease are perfect mirror images of each other. It just makes our comparisons way more fair and accurate.
But we're not out of the woods yet. Even with clean signals, you can have these system-wide errors. Maybe the red dye was just a little bit brighter than the green one. Or maybe one part of the scanner was slightly out of focus.
These are what we call systematic biases, and they can skew all of our results. It's like trying to take a level picture with a tilted camera.
And this is how we fix that. It's a process called normalization. Before we do anything, you can see the data is all skewed and off-center. That's the bias we were talking about. Normalization is basically a statistical way to digitally straighten that tilted picture. It recenters the whole dataset. We've leveled the playing field, making sure that any differences we find from here on out are real biology, not just some technical quirk.
All right, now for the really exciting part. Our data is clean, it's reliable, and now we can finally ask the million-dollar question. Which specific genes are acting differently between our two samples? It's time to find our prime suspects.
But think about it. This is a huge challenge. We've got data for thousands of genes. Some of them are going to look different just because of random chance or tiny measurement errors. So how do we figure out which changes are real and which are just random flukes? Just picking the ones with the biggest change isn't scientific enough. We need proof.
And the tool we use for that proof is the p-value. To put it simply, a p-value tells you the probability that what you're seeing is just a random coincidence. So, a really, really small p-value means it's super unlikely to be random, which gives us confidence that we're looking at a genuine biological effect.
For instance, if a gene gets a p-value of 0.01, what that's telling us is there's only a 1% chance, just 1 in 100, that the change we saw was a fluke. So scientists will set a cutoff, usually something like 0.05, and any gene with a p-value lower than that gets the label statistically significant. It's basically our stamp of approval.
And this is the payoff for all that work. This table is showing the top 5 genes that were flagged as different in a study on zebrafish. You've got the gene's name, the log fold change, which tells you how much its activity went up or down, and then that all-important p-value. Just look at how tiny those numbers are. This is it. This is our most wanted list of genes that are almost definitely playing a key role in whatever biological process was being studied. Okay, so identifying individual genes, that's a huge win. But the real magic of this technology comes when we step back and see how all these different genes are working together. It's time to zoom out, see the bigger picture, and check out the incredible payoffs.
This quote just nails the shift in thinking. You know, old school genetics often focused on one gene at a time. But genomics, which is what we're doing here, is all about understanding the entire network. It's about figuring out how whole teams of genes work in concert to create a result, whether that's a healthy person or, unfortunately, a disease.
And one of the coolest ways we see these networks is through a technique called clustering. We basically tell a computer, hey, find all the genes that are acting alike across our samples and group them together. This analysis of leukemia patients is just a mind-blowing example. Each column is a different person, each row is a gene, and the colors tell you if the gene is on or off. And look, just based on those gene patterns, the computer was able to perfectly split the healthy people from the leukemia patients. They formed 2 totally distinct groups, revealing a clear, undeniable genetic signature for the disease. And that brings us to the so what. Why does all of this matter? Well, because these genetic signatures are unbelievably powerful. We can use them to diagnose diseases way earlier and more accurately. We can classify different types of tumors to make sure a patient gets the best treatment for them. We can even start to predict if a person will respond to a specific drug, which is the whole dream of personalized medicine. And of course, we can discover brand new targets for the drugs of tomorrow. So what started as a pretty glowing image on a little glass chip ends up being this profound new way to understand biology and to fight disease. We've basically learned how to listen in on the incredibly complex conversations that our genes are having all the time. The only question left is, what disease should we listen in on next?